everybody. We're going to walk through the lesson today. It is February 2nd, and so today is a great time to start your lesson if you haven't already. This week, what is due is the energy challenge for respiration and photosynthesis. So we're going to walk through this lesson together, and if you do it with us with the video guide, hopefully you'll get all the points. And we're also going to talk about how to take notes. So Emery had a great question this week, and she was wondering, how do we take better notes? We had Tiffany who taught us how to take notes last time, and so we're going to take notes along the way as we go through this program. And that's what we're going to do. So are you ready? Here we go. All right, so we're starting with the energy challenge respiration. Here we go. All right, and I'm going to go through and read all of the words for you, and that's what I would suggest you do in these programs. If you slow down and just take a minute to read it all, and at the end then we'll take some notes from it. Okay, so introduction, energy challenge. We all know that we need to eat to get energy, but how does the food we eat power our bodies? In the second lesson in the Gathering Energy series, we'll investigate just how that happens. On a molecular level, it's just a swirling mess of molecules, some long, some short, each with different elements, and yet our bodies and other living things have an advanced way of taking energy from food, storing it, and using it later. These processes in our body take place all the time, millions of chemical reactions every second. You're going to play the energy challenge game, where you'll generate ATP molecules by running digital versions of biochemical pathways. And here it says a biochemical pathway is just a sequence of chemical reactions. Through the game, you'll explore just how we get energy out of the food we eat. And so up here in this picture, we see two people it says fill up on salad. How does the food we actually eat fuel our bodies? So you might look at that salad and think, okay, there's some glucose, some cellulose from the lettuce. Maybe there's some fructose if there's any fruit on there. All of those are carbohydrates. What other macromolecules could you put in a salad? Maybe if you put an egg on there, you'd have some protein and some cheese, you'd have lipids or fats. And then anything that was living will have DNA in it. If you've ever isolated DNA from strawberries or peas, in class you'll know that. Okay, so from this, our learning objectives are to be able to explain and order the process of aerobic respiration and account for the molecules consumed and produced. And we're also going to be able to do that for the process of fermentation or anaerobic respiration, which is when you don't have oxygen here. So anything with an A, remember from our live lesson, A like asymmetrical means not or without, so without oxygen. We're going to be able to account for the molecules consumed and produced. So I'm going to pause here. Okay, so can you see my screen here? On my notes, I just took those two learning objectives and I divided my paper in two. So the top is aerobic respiration, and I put two boxes with an arrow and then two boxes on the other side. And this is the basis for a chemical reaction. This is a model that's going to help us represent what we learn, and what we learn through words we're going to be able to diagram with pictures. So this is a great note-taking tool. To be able, if you're a super visual learner like I am, I like to see it diagrammed out. So in both my boxes, those are the molecules that are going to be consumed or used. You might also know the word reactants if you've taken chemistry. And then our products, or the things produced, are the ones that come out on the other side. And you read it left to right. And then I left some space so we can enter in some information. And then at the bottom here I put anaerobic respiration, which they also called fermentation. And we have the same process. Two boxes in, two boxes out. Okay, so if you're taking notes along the way, go ahead and set up your paper like this. All right, so here we're going to learn what goes in and what goes out by analyzing this graph. So the text says, In the last lesson, we looked at cellular respiration, the process by which cells get energy. Now let's take a closer look to see how much energy can be obtained from glucose and get a sense of the important details in the process. The graph to the right represents the process of cellular respiration in a eukaryotic cell. That's like a plant or an animal cell, things that have a nucleus. Read the graph, then mark whether each molecule listed is increasing, decreasing, or constant over time, or not present. So as I look at my x-axis, that's time in seconds from left to right. That's the change over time. And then on my y-axis, I see concentration, which they measured in molarity. Okay, so I can see, let's just take a look at let's go here, carbon dioxide. That's the green line. And over time, I see that it is increasing. ATP, I see it goes from down to up. That is also increasing. Glucose started here, ended down here. I would say that that's decreasing. Nitrogen gas, N2. Do you see nitrogen gas on our graph? Nope. So that is not present. Water vapor, that's this gray line, goes from down to up, that is increasing. And oxygen goes up to down, that is decreasing. I'd like to review how to read graphs. So if you want to review how to read, should we do that? I guess we just kind of went over it. But if you want to go and review how to read graphs, click that box. Okay, so from that, let's pause there. Okay, so from that graph, I can actually fill out my first produced and consumed. If something was increasing, it would be produced, and if something was decreasing, it was consumed. So in our graph, we produced CO2, and I'm actually going to be fancy and draw the molecule out. C with 2 lines and O's on either side. Here's my CO2. And what else did we produce? That money-making molecule in my brain. Money is green. ATP is like the energy of the cell. We sort of say it's like money because you can use and exchange it. It's like having a bank account, so I'm going to put that in green. Okay, so now I have CO2 and ATP produced. And then what was consumed? Okay, so then to finish our diagram here, glucose goes in, and that's why it was consumed. It went down in the graph, and oxygen was also used and consumed. And we're missing one thing, yeah? So we need to add in water. So oxygen, we gotta add in a box, which is totally fine. So just add your box in. Okay, so I'm a little bummed I had to add that box in, but it looks great there, that's fine. But if you do want to set up your notes so that you can write underneath each one what it is, whoa, that would be really cool. So you can write more details underneath because we'll learn a little bit more here. All right, so let's go back to the program and see what else we can learn. Okay, so we're going to break it down now into different steps. The first step in cellular respiration is called glycolysis, where glyco equals glucose and lysis means to break down. Glucose is broken down part way in this first step. Let's take a look at some of the details. The graph to the right represents the process of glycolysis in a eukaryotic cell. Read the graph, then mark whether each chemical listed is increasing, decreasing, constant, or not present. All right, so we know how to do this. Carbon dioxide, not present. Pyruvate, increasing, and that was that molecule that we broke down glucose. Remember in the intro to the animal cell, we made pyruvate. Glucose is decreasing because we're breaking it down. Nitrogen gas, we're not worrying about. It's in the air, but it's not used in this process. And we're making some ATP, so that's increasing. And we're not using oxygen yet. So that is not present in this particular graph. Okay, so check it. We did it. Now let's make some notes about glycolysis because we can do the same sort of process here for that. Okay, I don't often suggest this because it takes a lot of time, but I did rewrite my notes because I wanted these to look pretty. Okay, so I'm also taking time to color coordinate. So I know, Emery, you had kind of said like, oh, color coordinating is kind of lame, but it's actually really awesome. I hope you can see this because it allows you to keep track of things. So every time I write glucose, I'm going to keep it in pink. And then when I dissect this word, the step for step 1, glycolysis, I can see glyco means glucose, I write that in pink. Lysis is to break down. So here now I have the same scheme, and I made my boxes. So I take glucose, and I make pyruvate and ATP. So I'm going to fill those in now, and I'm going to keep the color coordination. So ATP is always going to be green, and then pyruvate gets to be another color. Here we go. So I went ahead and I added in the chemical formula too. So you'll notice glucose is C6H12O6, and then pyruvate is C3H6O3. So what do we need to do in order to balance this equation? If we have 6 carbons on this side, because energy and matter cannot be created or destroyed, we didn't just like disappear half of this molecule. We actually made 2 pyruvates. So if I wanted to balance this equation, I'd write it 2 by pyruvate. All right, so here's where it gets fun, the energy challenge game. In this game, we'll use the biochemical pathways, or those series of chemical reactions, to generate a target number of molecule ATPs, and they're calling this the energy currency, or money, in living things. Some of these pathways take place in our bodies, and others don't. They take place in plants, bacteria, and even archaeans. Remember those extremophiles that live in Yellowstone and the Antarctic? Prokaryotes that are like bacteria. Pay attention to the ingredients you're adding. Those are like the things that are consumed or the reactants, the chemicals that come out of the pathways, the things that are produced, and amounts of each. There will be questions about the details after the game, so this is maybe where we need to take notes on that. In the next screen, you'll see only one pathway, glycolysis. Your target is to generate 8 molecules of ATP. Glycolysis is the pathway you will use to generate ATP. It is represented by 2 arrows. And maybe the 2 arrows are because you're taking 1 glucose and making 2 pyruvate. And then this blobby thing is ATP. Hit the X on the top right-hand corner to close the informational panel and get started. So I go up here. If you see this again, hit the orange information icon. Okay. All right, so here we're in our pathway. What ingredients do we need? Drag a pathway here to get started. So let's drag this pathway there. Ingredients, what went in, right? We needed glucose, yummy, our sugar. And what else should we put in? ADP we need to put in because this is going to help us generate ATP. We're going to put 1 more phosphate on there. We're also going to need, we don't need oxygen for this, remember, but we are going to, if we're making ATP, adenosine triphosphate from adenosine diphosphate, we need a phosphate. And then we need our electron carriers. Do you remember? Those are like the buses that are going to help this chemical reaction move forward. Oh, nope, not that one. Okay, so a lot just happened there. Whoa, okay, there we go. So, okay, so that went really fast and a lot of stuff happened there. What we can do is we can slow it down and access an animation of it here. So if we watch this one more time, let's let it go ahead and play. I'll tell you what's going on here. We had a phosphate that we needed along with ADP. These 2 are going to combine together to make ATP. We had our electron carrier in an empty bus position. We had to put in some energy, ATP, to make it go, and we had glucose. So I'm going to pause it. That was a lot that just happened. Okay, so that was everything that went in. Pause. So now we're going to put energy into the reaction, that's the ATP. We're going to put glucose in. Pause. and we're going to put in our electron carrier. So when we break down glucose, an electron is released from the bonds between the carbon molecules, and that loads onto the electron carrier bus. That's why the electron went from glucose over to NAD, and it made a molecule of, there you go, NADH. So NAD plus, NADH. Think of the plus as like it was positively charged, and we added an electron, and that made it neutral. So that's where NADH comes from. Okay, so, and then we put an ATP and, we put an ADP and a phosphate, and we got out ATP. And then this number here shows how many pyruvate. So we had 2 pyruvate, remember, because we broke down 1 glucose, broke it in half to make 2 pyruvate. All right, so now we're going to look down here and summarize what we just learned. We had to put ATP in, and we got it out. Carbon dioxide, we didn't do anything with carbon dioxide yet. Remember, we just went from glucose as an input. NAD plus was our input. That was the empty bus position of the electron carrier. When we put an electron in, that was our output, NADH. We haven't used oxygen yet either. Remember, this is glycolysis. This is actually going to form anaerobic. This is the basis of anaerobic respiration. Pyruvate was produced. That was an output. And we didn't use water yet, so that was not involved. All right, we did that right. Good job, us. All right, so here we go. Enter the following questions based on your observations about glycolysis in the energy challenge game. If you'd like to see it again, we can watch it again. But let's see. Okay, yeah, so now we have to look quantitatively. We're going to see how many of each were formed. And we have to do some math. So if 10 were made and 3 were used, that would be 7. Okay, so let's watch it one more time and we'll keep track here. And we're looking specifically at ATP, glucose, NADH, and pyruvate. We put in 2 ATP. And we got out 4 ATP. Did you see that? Let's go to the end. Let's pause. 4 right there. So if we put 2 in and we got 4 out, how many total did we get? 2 that were produced. Glucose, we put 1 in and that was consumed. NADH was produced. Here you can see, and then it has a number 2. 2 were produced, and pyruvate, 2 were produced. All right, we did it. So I'm actually going to add that into my notes as well. So I added in, here I am, I added in 2 pyruvate. I'm going to add a 1 next to glucose. And I'm going to add a, actually now I can even add in, plus we added an ATP at the beginning. I'm going to pause this. All right, so my new updated glycolysis reaction looks like this. Oh, sorry guys, I really want a document camera, that would be cool. Okay, so 2 ATPs went in, 4 ATPs came out, 1 NAD plus went in, 1 NAD H came out. Or wait, what did we just say? 2. 2 went in, 2 came out. And that's because we're breaking down 1 glucose, those 2 electrons going there, 2, 2. Okay, this is all like a lot of details, but just it's a good process to go through what goes in and what goes out, what's consumed, what's produced in each one of these chemical reactions. All right, what do you think is responsible for the points on the graph where you see a small decrease in the number of ATP? Let's read through our options. ATP is unstable and disintegrates on a regular basis. Eventually, more gets made. That could be an option. ATP is produced as the first step in glycolysis and later it is used. ATP is being used in the first step of glycolysis, then more ATP are being produced in the second step of glycolysis. Or is it that natural fluctuations in the experimental process cause the ATP to go down and up? So from these four options, the one that we just saw happening was this third one. ATP is being used. Remember, it went in to the equation, and then we actually produced more ATPs afterwards. All right. We will focus on what glycolysis does for the cell and what it uses and produces. The energy challenge game you are about to play uses icons to represent the molecules and processes involved. Shown to the right are the structures and icons for the major input, output, and pathway. Which is the smallest molecule shown on the screen? Which is the smallest molecule shown on the screen? I would go with pyruvate. Glycolysis is a process. Pyruvate has less carbons, less hydrogens, less oxygens than glucose. What type of biological macromolecule is glucose? Remember, that's a carbohydrate. It's like a sugar. Cool, we did it. All right, next slide. Components of glycolysis. You will focus on what glycolysis does for the cell and what it uses and produces. The energy challenge game you're about to play uses icons. Shown to the right are the icons for the electron carrier molecules. Those are like our electron buses. We're calling them buses because they take on electrons, and then when it's their stop, the electrons get off. Okay, so to continue, enter the symbol of the element that carries the positive charge in NAD+. The element that carries the positive charge. So we're looking at this element, and I see next to oxygen, there's a little minus sign. Next to nitrogen, there's a little plus sign. So the molecule that carries the positive charge is nitrogen. Incorrect. Take a closer look. Maybe, oh, here we go, the symbol. So you want to enter N. There we go, we did it. N for nitrogen, just enter N. You will focus on what glycolysis does for the cell and what it uses and produces. Okay, so this is the same paragraph each time, it's lame. But anyway, what is the major difference between ADP and ATP? So here we have ADP, adenosine diphosphate, only 2 phosphates. And then ATP has 1, 2, 3. So the difference is 1 phosphate. What is the total charge on the phosphate ion? Enter the charge as the symbol and then the value. So the symbol would just be PO4. That's because it has 1 phosphate and 1, 2, 3, 4 oxygens. And then how many negative signs does it have next to it? 1, 2, 3. So this is PO4 minus 3. There were some issues. Okay, let's figure out what exactly it wants us to do. Maybe don't put a space. Okay, let me pause. Okay, this is why it's beneficial to watch this video with your teacher making all the mistakes. So I read this wrong, as maybe is easy to do. All it wants us to do is put in the symbol, the minus sign, and then the number. Let's see if that's right. Yes, we did it right. All right, next graph. All right, here we go. Pause and reflect. We just spent some time learning about this. How well do we understand this? I get it pretty well. I hope you do too, especially if you took notes. Now you can look back at your notes and you can see, here I am, there's our notes. You can see this process very colorfully recorded for yourself. How well do you understand glycolysis? I feel like I get that pretty well. Maybe you're still confused about the NAD pluses and NADHs, but again, Those are just little electron carriers, the buses that help move our electrons around, which is going to be important in our next step. Okay, I want you to add in one more piece to your notes, and that's under ATP. This is really important. We go from ADP with 1-phosphate to ATP. So add that in underneath your ATP. That's the major step there. All right. Now we're going to choose our path, oxygen or no oxygen. So go ahead and take a deep breath in and breathe everything out. So every time you breathe, you're sucking in air around you, and that's 21% of that is oxygen. There are some environments, though, that don't have oxygen. So specifically here, they're showing us in a designed anaerobic pond, there might not be any oxygen. There's also lower oxygen in swamps. Swamps don't have a lot of oxygen, and there's a reason for that. But let's go ahead and read the paragraph. It says, the amount of oxygen in an organism's environment is important. It determines which options are available for how living things will be able to gather energy. In general, aerobic processes produce more energy than anaerobic processes. Anaerobic processes take place without oxygen. As a result, only anaerobic options such as fermentation are available. Aerobic processes take place using oxygen. With oxygen, the Krebs cycle and aerobic electron transport become options for organisms that can use them. Glycolysis is neither aerobic nor anaerobic. This is unlike most other processes. That was kind of a confusing paragraph, but Okay, so select an image below to choose an environment to investigate first. Let's go with the oxygen-rich forest. It says here the second pathway in aerobic cellular respiration is the Krebs cycle. It comes right after the first pathway of glycolysis, what we just learned about. So here's the Krebs cycle. The second step in cellular respiration is the Krebs cycle. In this cycle, there are a series of reactions that occur in the mitochondria. Remember, that's the powerhouse of the cell, right? That's where we break down pyruvate to make energy, or ATP. If oxygen is present, pyruvate from glycolysis can enter the Krebs cycle. The first input into the Krebs cycle is acetyl-CoA, a molecule very similar to pyruvate. To be honest, these are details that you probably don't need to know at a 9th grade biology level. This is more so in college. So, but we're gonna work through this. It'll be good. The graph to the right represents the Krebs cycle. Read the graph, then mark whether each chemical listed is increasing, decreasing, constant over time, or not present. So we know how to do this. CO2, increasing. Acetyl-CoA, that's this blue line. That's like pyruvate, that's decreasing. Glucose, do we see glucose here at all? Nope. Again, nitrogen, it's there, but we don't care about it. ATP is increasing, and I don't see oxygen. Nope. Not present. So from here we can take some notes on that. Let's go ahead and do that.